Hello, everyone. Welcome back to part two of our lecture on the cell. So last time we talked a little bit about what cells do, how we look at them, and we talked in some detail about the plasma membrane. Now we're going to dig a little bit deeper and talk about structures beneath the plasma membrane. So that brings us to the cytoplasm. So the cytoplasm is everything in between the cell membrane and the nucleus. This will include the cytosol, the sort of liquidy soup part of the cell, of the cytoplasm, inclusions, just masses of material that have been clumped in this area, and organelles, the little structures where the cell can subdivide its biochemical machinery. So first we'll talk about cytosol. As I mentioned, viscous fluid containing lots of stuff. This will include ions, electrolytes, sodium, potassium, calcium, all that kind of good stuff. Nutrients, things like vitamins, glucose, energy source components for the cell. You'll have various proteins floating around in the cytoplasm. Carbohydrates, which I kind of already talked about. The sugars, lipids, fats floating around that can be used for energy or to serve as cofactors, and numerous other small molecules floating around the cytosol. But the main component is most of these things are going to be water soluble, so they're in solution suspended in the cytosol. Now, inclusions, these are just areas where large amounts of material are tightly packed together to form an aggregate. Two good examples of these are melanin, which is the pigment in our skin that gives our skin color, or the primary pigment that gives us color. So you can find melanin in inclusions, or glycogen. Glycogen is a polysaccharide that animal cells use to store energy. So we have glucose floating around. Well, you just take glucose molecules and stick them together in a very particular fashion, and you're going to make these large spiral aggregates called glycogen. What is it? It's just a whole bunch of glucose molecules stuck together. When your cell needs some energy, an enzyme comes along, just cuts off one of those glucose molecules, and it can get burned for cellular metabolism, or specifically catabolism. All right. Organelles. Organelles means little organs or small little machines inside the cell. A lot of different kinds, and we're going to talk about most of the important ones. This is useful because it allows for the division of labor and the establishment of special chemical environments where particular reactions can happen most efficiently. This is one of the major differences between eukaryotic cells, meaning cells like us, and prokaryotic cells, meaning things like bacteria. Bacteria don't have these little organelles. Eukaryotes, true eukaryotic plants, animals, we do have these organelles. That allows the cell to be much larger and carry out much more complicated processes. Two basic divisions of organelles. We have membrane-bound organelles and non-membrane-bound organelles. So what we mean by membrane-bound is it has another cellular membrane. So it's got a very similar structure to what we saw: a cell membrane, a phospholipid bilayer. Here, membrane-bound organelles have their own phospholipid bilayer separating from the rest of the cell, which again just allows for the establishment of a particular environment that's specialized for one particular job. So, membrane-bound organelles, right? Biochemical activity in each organelle is isolated from the cytoplasm and other parts of the cell. Examples of membrane-bound organelles: endoplasmic reticulum, the Golgi, lysosomes, peroxisomes, and mitochondria. So let's just take these one at a time. So endoplasmic reticulum, a network of interconnected membrane-bound tunnels. So these spaces inside these structures are called cisternae. So a cistern is just an underground chamber to store water. So people used to have clay pots buried in the ground, and that is a cistern. It's just where the term comes from. Two types of ER, smooth and rough. Smooth ER does not have ribosomes stuck in it, and rough ER does have ribosomes kind of peppered in there. So this shaded out section, the area that's grayed out, that is the nucleus. Immediately surrounding and connected to the nucleus we have our rough ER. Rough ER. Outside of that, we're going to have the smooth ER, where we are not going to have ribosomes. So these little folds, these structures are made of phospholipid bilayers, both smooth and rough. The rough ones simply have ribosomes here. You may remember that the nucleus contains the genetic material, and the genetic materials is going to code for the various proteins the cells are going to make. So it kind of makes sense that inside the nucleus, where you store the information, right next to it are going to be the protein factories for producing protein. Okay. Smooth ER, right? So this has, it is connected to the rough ER, right? It is also continuous. So there'll be some connections right here. So the smooth ER really has a specialization in the synthesis, transportation, and modification of lipids, including things like hormones. It's also a really important place for the breakdown or metabolism of carbohydrates. So carbohydrates and lipids are the specialty of the smooth ER. It's also where the cell is going to detoxify a lot of chemicals, things like drugs, alcohol, poisoning, right? Your cell can break down a lot of these compounds here in the smooth ER. In fact, if people chronically abuse alcohol, you can actually see it in their cells. The proportion of smooth ER to rough ER will be different, right? So people who chronically use alcohol, right, they're going to have a greater proportion of smooth ER in their cells because their cells are trying to detoxify this compound. Rough ER is rough because it has ribosomes attached to it, right? This is intimately involved with the production and then transportation of protein in this area. So for reasons I'm going to explain in a second, but if you want to make a protein that ends up on the surface of the cell, if it's going to go to a organelle, or if it's going to be secreted outside the cell, that protein must be made inside the rough ER. So if you don't want the protein outside the cell, if you just want it floating around inside the cytoplasm, it's going to be made by a different ribosome. It's going to be made by what's called a free ribosome. So a free ribosome is floating out in the cytoplasm. That's where you make your protein. That's around the cytoplasm. But if you make your protein in the rough ER, then you can take a little bit of the ER and pinch it off to make a vesicle, and then the body can send that vesicle to different places. Usually it goes to the Golgi, but we'll talk about that once we get to the Golgi. So that's why the rough ER is very important for secretion inside the cell. If you want to produce something that gets secreted, which means released from the cell, you got to make that in the rough ER. Right? Incorporated into the plasma membrane, right? Those two things are going to be related. If it's going to go to the plasma membrane on the outside of the cell, right? That's how it's going to get there, through the rough ER. It's also important for the creation of lysosomes, which we'll talk about in a little bit. Okay, so the rough ER is right outside the nucleus. So if you need to make something that is transported anywhere, it needs to be made inside the rough ER so it can be transported to the Golgi. Now, the main function of the Golgi apparatus is sorting and transportation. Think of it like the FedEx hub of the cell. The Golgi is going to be receiving materials from the rough ER, and it's going to say, this particular protein has this little barcode, this little marker on it, and that means it needs to go to the mitochondria. So it will send a little vesicle delivering that protein to the mitochondria. Or it'll read a tag to say, this needs to go to the plasma membrane, and it gets sent to the plasma membrane. Or another little tag will say, okay, this has to be secreted from the cell. It'll be sent to the plasma membrane and then released to the outside of the cell. So the main function of the Golgi apparatus is sorting and transportation of materials from the rough ER. It has a similar looking structure to the ER, so you're going to have these little folds of phospholipid bilayer. The spaces inside these folds are called cisternae, but the Golgi is a little bit smaller. It's a little more compact. It looks like usually looks like a small stack of pancakes. It has two faces. It has what's called a cis face and a trans face. The cis face, cis means same. So cis means the same side. It's the side facing the nucleus, the side facing the rough ER. Trans means opposite or on the other side. The transphase would be on the other side of the nucleus. So this is what the Golgi looks like in a transmission electron micrograph image. Over here, we see the general layout of the cell, nucleus, rough ER that has been grayed out, and then the Golgi here is in green. So take a zoom in here, and you can see that cis face of the Golgi receiving vesicles filled with proteins created in the rough ER that get sent to the cis face, the side that's facing the nucleus. Right? It will go from one pancake to another, where you can have modification and packaging and things like that. And then eventually it will be set to a vesicle, which is then sent to wherever it needs to be. Oh, hold on just a moment. Again, sorry about that minor little hiccup there. Okay, so we left off talking about the Golgi apparatus. Material produced in the rough endoplasmic reticulum is sent to the cis face. There it will be modified and labeled as it passes through the Golgi. When it gets to the trans face, that material will be packaged up in a little vesicle and then sent to wherever it needs to go. Right, and that's just basically what these next few slides describe what I just talked about. Right, protein synthesized in the rough ER gets packaged into vesicles. Vesicles bud off from the ER, they go to the cis face. They move through the various cisternae of the Golgi. Once they get to the trans face, they're packaged into other vesicles and sent to wherever they need to go. If you want to look at this process in a little bit more detail, I have included this figure here. You can see it broken down. Again, this is not a cell bio class. I just wanted to give you guys a little bit more information should you want to investigate. Again, mostly, I just want you to take away what are the various structures, what are the various organelles of the cell, and have a general idea of their function. You don't need to go into excruciating detail about exactly how the Golgi works, but it is very interesting. Okay. Next organelle, lysozymes. So lysozymes are generated from the Golgi. They're going to contain various digestive enzymes. So what these are, think of these as the garbage disposals of the cell a little bit. If anything the cell wants to destroy, it can combine with one of these lysozymes and release these digestive enzymes to break down whatever materials are out there. A good example of this, perhaps you've heard of a type of cell called a phagocyte. Phago means to eat, so a phagocyte is a eating cell. It's a big eating cell. So when a cell, say, gobbles up a bacteria, right, it brings the bacteria inside of itself, and the bacteria is just sitting there, doesn't really care. So what the white cell will do is take one of these lysosomes and fuse it to the vesicle containing the bacteria that releases digestive enzymes that then breaks down the bacteria and destroys the bacteria. Now, sometimes these lysosomes are used to break down old or worn out organelles. Say like you have a mitochondria that's kind of old or worn out and broken down, you need to break it down. So you can just take that thing and add a lysosome to it, release the digestive enzymes which break down that organelle so you can recycle its components. Now, another thing that lysosomes are capable of doing is this thing here called autolysis, also called apoptosis. So if a cell just opens up and releases the contents of all of its lysosomes all at once, enzymes that break down carbohydrates, that break down lipids, that break down DNA, that break down protein will actually just digest and completely destroy the inside of the cell. And you might say to yourself, good Lord, why would you ever want to do that? Right? Why would you want the cell to commit suicide? Well, remember that I talked about this is an excellent way to prevent cancer, right? If a cell starts determining, it starts figuring out that it's losing control of its cell cycle, it might be precancerous, right? It's to the benefit of the entire organism that cell destroys itself. In fact, there are many processes in our cells that prevent cancer in exactly this manner. Once they detect certain problems, a cell just commits suicide in order to avoid becoming a tumor. There's also a constructive reason why you might want to do this apoptosis or the autolysis. You guys think of an idea of how you could make something by destroying something? Well, I got 5 of them right here. And if you've ever seen a picture of a developing embryo, right, digits are webbed, right? And at a very particular point of embryological development, all these tissues, which are all grown together, all the cells in between these 2 fingers die away. They undergo programmed cell death. They undergo apoptosis, which is the reason why I have 5 distinct digits here. If that didn't happen, you would have massively webbed fingers and massively webbed feet. And that sometimes is seen when that process does not occur. So I just want you to know that this is also important for embryological development, for shaping certain structures. It's not just the growth of certain things, but also the paring down or the removal of certain cells as well. Again, this is not an embryological class. This is not a developmental biology class, but I do want you to know that these structures do play an important role in making or in playing a role in creating the final structures we see as adults. But I wouldn't test you on that. Okay, this is what the lysosomes look like. Small membrane-bound organelles filled with lytic and digestive enzymes. Peroxisomes. Now, peroxisomes look kind of similar to lysosomes. They tend to be a little bit smaller. They're also made, or they are made as little offshoots of the endoplasmic reticulum. And here they're used to detoxify what are called reactive oxygen species. So to give a little bit of background on this, oxygen is an extremely, or molecular oxygen, O2, is an extremely reactive compound. In fact, when oxygen was first created on Earth, it was a catastrophe. When plants, or I should say cyanobacteria, started making oxygen for the first time in large concentrations, it was a catastrophe. It led to a mass extinction that literally wiped out most of the life on this planet. Right? Oxygen is very, very reactive. It completely changed the chemistry of Earth. To kind of give you an idea of how basic a shift this was, before plants made oxygen, the sky was red. It was not blue. It was actually red because of methane. Plants started producing oxygen. Oxygen is a very reactive compound, starts reacting with the methane. The methane basically eventually ends up going away. We get an atmosphere that's predominantly nitrogen, which is why we have a blue sky. If you want to know more about that story, ask me during lecture, or ask me during office hours, but oxygen is a big deal. It's a highly reactive compound and it actually damages cells, right? And you might think to yourself, that's kind of crazy, right? If we don't get oxygen, we die. And that's true because our cell's physiology is learned to use this highly reactive compound to help liberate energy, right? We do that in the form of metabolism. But in doing so, we generate dangerous compounds, right? Toxic side compounds. And our cells have to have a way of dealing with this. And how do they deal with these reactive oxygen species, these toxic reactive oxygen-based compounds? Peroxisomes. So when you live in oxygen and when you breathe and when you do metabolism, you're going to make toxic side effects. You know, it's really true. Food is poison. The more you eat, the more reactive oxygen species you make, the more likely you're going to get cancer or something like that because you're damaging your cells. Look at me. I'm obviously not making any sort of social commentary or judging people on what they do, but it's really a thing, right? You guys probably haven't heard of it in this particular context, this particular perspective before, but I'm sure you've heard of antioxidants. Right? Antioxidants are good, right? Everybody wants their antioxidants. Well, this is what this is all about. Your cells, since we use oxygen, we have to get rid of these reactive oxygen species. The way they do that is through peroxisomes, and particularly these enzymes inside our peroxisomes. Enzymes like catalase, superoxide dismutase. You don't need to know these. I'm not going to test you on it, but I want you to know there's a host of enzymes inside the peroxisomes which remove these toxic versions of oxygen. They need certain cofactors, which are the antioxidants. So if you want to stay healthy, you take antioxidants into your diet. Why? Because it helps you make these enzymes which remove the toxic oxygen species. Where are these enzymes found? inside peroxisomes. So, long story short, take your antioxidants, make your enzymes like catalase, peroxidase, superoxide dismutase. This keeps your peroxisomes happy. Happy peroxisomes keep toxins from building up inside your cells. You don't get cellular damage, you don't get cancer. So that's really the whole long story there. Okay, back again. So yes, peroxisomes. These are organelles specifically designed to help detoxify reactive oxygen species. More of these you got, the healthier your cells will stay. Just what they look like under a transmission electron micrograph. Okay, mitochondria. These are perhaps a little bit more familiar to people. Bean-shaped organelles, they have a double membrane, right? The inner membrane is folded very highly into structures called cristae, and inside we have a material called matrix. So here's where the vast majority of ATP, which is the energy molecule of the cell, adenosine or adenine triphosphate. This is going to be the energy currency of the cell. Energy will be stored in the form of ATP, and the cell can use this when it needs to carry out some particular process. Job of the mitochondria is to contain the electron transport chain that is going to be responsible for making all this ATP. So here is the outer membrane of the mitochondrion. Here's the inner membrane folded into structures called cristae. Inside the inner membrane, we have the matrix. Now, if you look at the fold of the cristae, you're going to get this blow up right here. So these little lollipop little structures here are called ATP synthase. So these are actually membrane-bound enzymes stuck in the membrane. These proteins, these enzymes are going to be synthesizing ATP. They're going to be regenerating ADP into ATP and producing energy for the cell. That's why they are the powerhouse of the cell. Now, taking a look at one of these things, one of the things I usually ask my class is, what does this look like to you? Right? What does this little organelle look like? And most people will say, oh, it looks a little bit like a caterpillar. It looks a little bit like a worm. Some of the students who've had microbiology before will look at this and say, it actually looks a little bit like a bacteria. And they'd actually be right. So I'm going to have a little bit of a side story here. I'm not going to test you on this, but I want to talk about something called the endosymbiont theory of life. So it is believed where we came from, where eukaryotes came from, long ago, like something like 3 billion years ago, you had some very large cells that were good at protecting themselves, but they didn't really make a whole lot of energy, and they were very, very simple. And you had some newly evolved, very, very small forms of life, a different kind of cell that had certain specialized metabolic capabilities. It had this stuff here. It had the ATP synthase, and it had the electron transport chain. So you had these little bacteria-like structures that were able to make tremendous amounts of energy. And so what you got is a symbiosis between the two. The small little cell that couldn't defend itself very well, but made massive amounts of energy, started living inside the larger cell. So the larger cell protected the smaller cell in return provided free energy to the larger cell. And that's the theory goes. This is how eukaryotic cells developed. This was once a free-living bacteria-like structure that was subsumed inside a different cell. And that's where we came from. Two separate cells joined together. And you might say to yourself, this is kind of a bold statement to be making. What is the evidence to support this? Well, there's actually a fair amount of it. One of them is, unlike the other parts of your cell, these mitochondria will actually divide like bacteria, right? They get bigger, they cut themselves in half, right? When the cell divides, it just so happens whatever mitochondria on the right, they go with that daughter cell. How many ever on the left, they go with that daughter cell. Also, you may have heard that the nucleus contains all the genetic material of the cell. That's not actually not exactly true. There is DNA found inside the mitochondria and nowhere else inside the cell. You can actually sequence the DNA in that mitochondria, and you can actually find similarities between free-living bacteria today, right? You can see they have a common evolutionary ancestor. So it reproduces like a bacteria, it has its own particular genome, and there is other evidence I don't have time to really go into right now. This was once a separate free-living cell that took up residence inside a larger cell. And then eventually those cells lived to learn in community. That's how we got multicellular organisms. There's actually the same story, this endosymbiotic theory, for chloroplasts, were once separate free-living cells capable of photosynthesis. They took up residence inside larger cells, and that's where plants came from. So, some other little interesting factoids here. Mitochondria tend to only be passed down mother to child. Just recently, they found that in some rare cases, it's possible for fathers to pass down mitochondria, but don't worry about that. Generally speaking, only the mother passes down mitochondria. And if you think about that for a second, it kind of makes sense. Because an egg, right, women carry eggs, they carry ova. Ova is an entire complete cell, mitochondria, you know, ER, all that kind of stuff, right? When the sperm enters the egg, the only thing that actually enters the egg is, in most cases, is the father's genetic material. With all the cellular components and half the genome from mother. When the sperm does its thing, it's going to release the other half of the genome, half from the mother, half from the father, put them together, you get 23 and 23, you get 46 chromosomes. But the mitochondria generally are only going to be passed down from the mother. So you can actually look at maternal relationships by looking at the mitochondrial DNA. And what I'm driving at here is something you probably have heard of, something called genetic Eve. Meaning is when you compare the mitochondrial DNA across all human beings on the planet and you work the clock backwards, you find that all human beings on the planet are descended from a single individual, right? Every human being on the planet is derived from one single individual that lived in sub-Saharan Africa like 120,000 years ago, right? So in a very real sense, we are all brothers and sisters of the same mother. One human being one woman gave rise to the whole human race. Now, as far as is there a genetic Adam? You can't really tell, right? We don't really know if that woman had one partner or multiple partners. We really, the genetic evidence isn't really there to say it conclusively. But it does pretty clearly show all human beings are derived from a single individual living in sub-Saharan Africa, something like a couple 100,000 years ago, which is an interesting fact. But more on that later. Okay, so now that we've wrapped up mitochondria and the interesting little fun fact of the endosymbiotic theory and the fact that all human beings are related to each other, that's a nice little place to stop. We've been going for a while. So we're going to come back in part 3 and finish up this section. All right, take care and see you soon.